Welcome to our second sensory session of 2023. Today we're going to be talking about Saccharomyces uvarum. In particular, we're going to be talking about the chemical and sensory properties that are produced when the particular strain of uvarum called QRHO is utilized in various different circumstances. But because Saccharomyces uvarum is a little bit new to us, I'm going to give just a couple of slides of introductions so that we all know what we're talking about, and then we'll jump right into the discussion of the wines that we sent to you through UPS. So, oops, there we go. So it's only recently that we've kind of gotten an understanding of all the microbes that are coming in on our grapes. We have to remember that most of these things look exactly the same under a microscope, or fairly much the same. So it's only really in the last 20, 25 years that we've started to get a really good view of all of the microbes that are involved. When we started to look at all of that, of course, we have all of these non-Saccharomyces yeast that we've started to hear about that are coming in. In fact, most of the microbes that come into the winery on our grapes are not Saccharomyces at all. They're made up of all of these other non-Saccharomyces species. And all these non-Saccharomyces species are thought to give us a lot of complexity in our ambient fermentations and even in some of our non-ambient fermentations. But they've also really provided strains or examples of things where enological companies have come and really, taken one strain of that and raised it up or selected it through the laboratory to be able to have selected strains of these two for various particular reasons. So for example, Mechnicauia or Torulaspora, these are things that, most of our enological companies at this point have some strain of those for either for bioprotection or for added complexity and that sort of thing. The other things that we've started to learn from this is what's happening during fermentation to the various microbes along the way. So again, we know that we've got this microbial community that comes in on the grapes. It turns out that what happens at cold soak, we have a different microbial community already by cold soak. And as soon as fermentation kicks off, we have yet another kind of microbial community. Really, it's getting much less diverse at that point, and we tend to have dominant microbes come out at that point. And that's generally where we see our Saccharomyces cerevisiae kind of overwhelming the population. But as we've sort of looked at more and more of these molecular techniques, a couple of things have come out. First of all, we've started to see that even throughout all of fermentation, not all of our non-Saccharomyces yeast go away. So for example, Hanseníaspora can sort of maintain in that population at low amounts and potentially have some impact on our fermentations. But the other thing that these molecular techniques have shown us is that not all of our Saccharomyces are Saccharomyces cerevisiae. So a lot of times we assume it's Saccharomyces cerevisiae that's doing this fermentation, but the more we've been able to look close into the genetics of these organisms, we realize that they're not all cerevisiae all the time. So for example, this data was taken from a Canadian winery. These are two different non-inoculated fermentations. And this particular winery, when they do their non-inoculated fermentations, it turns out that it's uvarum that is their kind of winery resident yeast population, and that it's uvarum that ends up doing the majority of their fermentation, not Saccharomyces. Another kind of zoom in on that shows you this is that same data, but just zoomed in to say we do have some Saccharomyces cerevisiae in there, but again, most of that is uvarum. So who is this uvarum, and how is it working? What does it look like relative to the other Saccharomyces that we know? And this slide just basically shows you what we currently know about the classification of uvarum. And what you can first see is that the classification of Saccharomyces, the whole clade of Saccharomyces, has changed considerably since the late 1990s. Again, that's when we started to have the tools to really be able to pick these apart with molecular techniques. The way that it sits now, what it looks like is that uvarum is really pretty far removed within the clade of Saccharomyces from Saccharomyces cerevisiae. So they're like third or fourth cousins along the way. But the other thing to realize is that uvarum's not new. It's something that's been around all along. We've just sort of been calling it something else because we couldn't tell the difference. So in the past, we might have called it Bianus, or we might have called it Saccharomyces Bianus variation uvarum, or some things like that. So again, this is something that's been around for a while, but now that we have a better way of telling who's who, we can figure out which characteristics belong to Uvarum and which characteristics belong to Cerevisiae in a much better way. So which characteristics do belong to Uvarum? It turns out, again, Uvarum is a fully fermentative yeast. It's been isolated from beer and wine and cider fermentations from around the world. They tend to be found mostly in cold regions. So for example, there's a winery in Alsace that their resident winery yeast is actually an Uvarum, not a Cerevisiae, but also in other cold regions. It's also found in tree bark. So the companies are sort of looking at tree bark at this point to say there's a lot of diversity of Uvarum over there. Are there other strains that we can raise up for particular reasons? But the overwhelming thing that we know about Uvarum is that it's really cryo-tolerant, meaning it ferments and it does really well at colder temperatures, which for folks that are doing cider fermentations or white wine fermentations at colder temperature, that could be an attractive quality. That same quality is why it's thought to sort of become the resident yeast at some of these cold wineries, and that it's able to overwinter in a way that maybe Cerevisiae just gets too cold and doesn't compete as well. But there are some really particular chemical and sensory properties that seem to be coming out from the various Uvarum strains that we've seen. So going back to that Canadian winery that sort of their resident population is Uvarum, they did look at different sort of the strain specificity of Uvarum or of these populations in 2 different years, in 2015 and 2017. And I show you this slide mostly just because I think it's very interesting. Most of the time we think with Saccharomyces cerevisiae that we have one dominant strain that's doing the fermentation. That does not seem to be the case with Uvarum, at least in this particular example. Here, what they were calling a dominant strain in this particular paper is anything that was more than 10% relative abundance in more than 2 of their replicate fermentations. So one of the most striking things here, I think, is looking at the majority of this is being, the majority of this fermentation is being done by what they're calling minor strains, meaning there's many, many, many different strains that are actually co-fermenting these particular wines, which makes you kind of just wonder if there's the potential for a lot more diversity as we learn more and more about Uvarum. But for today's experiments, we're focusing on one particular strain of Uvarum. So I guess that's something else to say that Vineyards has one particular strain that they have isolated from an ambient fermentation of Amarone wine. And then they've been able to sort of start to define some of the characteristics of that strain. But much like we see with Saccharomyces cerevisiae, as much as, for example, QA23 and DV10 and D254 all have kind of different properties, we would expect as we get more Uvarum coming up that those different Uvarum strains also have different properties. So part of what we're trying to do is identify which of those properties shine through in actual kind of production scale experimentation. So the one that we're going to be looking at today, all of our experiments today were fermented with a control and a control that we assume is Cerevisiae, compared to an Uvarum, this QRHO from Vineyards. So when Vineyards first brought this to us, the thing that really stuck out to me in these different characteristics of what Uvarum is doing is the ability to produce malic acid. So in some of their laboratory trials, they had found that instead of kind of consuming malic acid during the course of fermentation, it was actually producing malic acid. And that seemed like a really good potential way of building some acidity in some of our low acid varieties without having to just dump tartaric acid in there. But as we look at this list of distinguishing characteristics, we can see a couple of other things that are interesting. For example, Uvarum is thought to produce more glycerol. In fact, when I was at the wine board meeting a couple of months ago, Luca said that, in Italy, they look at Uvarum really as something to build mouthfeel because of this production of glycerol. The one thing that goes along with that a lot of times is less production of ethanol, because when sugar goes into the yeast metabolism, it can sort of get shunted off to glycerol or it can get shunted off to make ethanol. So oftentimes we do see a little bit less ethanol conversion as well. But then there's this other interesting characteristic right here, and that's these acetic acid numbers, as opposed to sort of producing between 0.1 and 0.9 grams per liter of acetic acid. At least their laboratory numbers show very low production of acetic acid, and that might either be low production or it might actually be consumption of acetic acid along the way, which of course would be very interesting as well. But one other thing about Uvarum to keep in mind is that it does have very different some different metabolism than what our Saccharomyces cerevisiae has. And so it does make some different volatile chemicals as well. And when we were kind of looking through the literature on this, and particularly looking through Enardis's in-house trials on this particular strain, one thing is more consistent than anything else in terms of these distinguishing characteristics of Uvarum, and that is a pretty high production of 2-phenylethanol. You can see that, here it's a really big increase in 2-phenylethanol. If you're not an organic chemist, 2-phenylethanol basically is a volatile chemical that is associated with kind of floral characteristics. So it gets descriptors like rose and hyacinth and just general floral character. So that's something else that we want to be sort of looking out for as we discuss the wines today. Okay, so with that kind of background on what Uvarum is and why we were interested in it, let's go ahead and jump into our experiments for the day. So the first flight that we looked at was an experiment that was done by Matthieu Finot at King Family Vineyards. So Matthieu, do you want to tell us a little bit about what your experiment was and why you were interested in doing this experiment?
Yeah. Again, after talking with Tyler from Vineyards, and he was I was kind of interested to try to play with it on Viognier. We always get very high pH, on Viognier, and based on what he was telling me, it was supposed to lower down my pH and increase my malic. So I'm like, if it's a way for me to not add too much tartaric acid, that would be a good thing. He was concerned about the very floral component that it could bring to the wine. I was not too worried about it because for me, I don't mind when Viognier is a bit more floral than too much of the apricots and peach flavor. So I was willing to try it on the Viognier just to see what it does. So that's the idea behind the experimentation.
Right. Yeah. So as Matthieu said, Viognier was a kind of a good option to start with this one for exactly those reasons. We tend to have higher pHs at harvest. And even if we kind of look at this is data, this is all of the WRE experiments that have come in since 2014. I just sort of put them on a graph, and you can see that, our Viognier pHs are quite high. The wine pHs are actually lower, but that's probably because, as Matthieu said, we're sort of putting a lot of tartaric acid in there to correct it. So is there kind of a way to get around that and do that correction in a more natural way? And again, the floral characteristic here might be good. We'll taste and figure that out. So Matthieu, tell us how you set up your experiment.
So it's a barrel fermentation Viognier, because it was the easiest way for me to make it work, to be able to have two controls and two experimentation. You've got the process here. There's nothing very special. Press, cold settle, and then racked into barrels inoculated with different yeast. We've got two sets of barrels that are identical. I'm not sure exactly which one we're testing here, but like we had two identical acacia barrels and two identical oak barrels. We might be testing the acacia barrels, but I'm not sure about that. And then after, just in the barrel room, monitoring the fermentation, we didn't add any acid at all, because the idea was really to not screw the experimentation by not adding exactly the right amount of acid. So by not adding acid, that was a good way to really see what it does to the wine. So
yeah,
as you can, my pH on this batch is great. Like everybody dreams of having 21.5 bricks with almost 4 pH on juice. It's beautiful. I love it.
Yeah, so it was a nice, this was good juice to start with, in the sense that you didn't need to add any sugar, you didn't need to add any nutrient. And for the case of the experiment, you didn't add any acid ahead of time. So thank you for your willingness to do that.
I don't know why I did that, but I did it.
Because I probably asked you to, so I appreciate it. So you used your control yeast in this case was Yeast Excellence STR. Do you have just tell us just a minute about why you like to use that particular yeast on Viognier, just so we know what you're comparing here?
Like STR, like ester. It's in the name. So ester is it's a yeast that produces ester, and so it's a yeast that I kind of like to use with La Motte Aviette. It's I feel like it's a strong fermenters. I mean, it's a no problem kind of yeast, and it works well for rosé and for Viognier, for that matters. I do use it. I used to use the GRE on Viognier. I've used the X16, and now I'm using mainly the STR. X16 is still good for me, but like the GRE, I can't do that anymore. It's too much stress for me. So it's never finished the sugar. And so anyway, so now I'm either X16 or STR on my Viognier. And the STR, I can use it in the in the tank fermentation, and also in the barrel. So.
Yeah, I wasn't familiar with this yeast. So as you know, I used to use GRE all the time when I was making back in the day. But so STR, I thought it was really interesting because it does have a lot of the same characteristics as what the description for Uvarum gives us. So it also is good at lower temperatures. It produces a lot of esters, and it even names some of the esters that we know that Uvarum overproduces. So I thought it was a really good choice in terms of a comparison for the experiment as well. Okay, so that you all know what you have in front of you. In your triangle test, of course, you had 2 of one wine and 1 of the other. So depending on what group you are, you can kind of look up your numbers here and see which is which. So again, the QRHO is our Uvarum yeast, and the STR is the Cerevisiae yeast. And I need to remember to be careful to say Cerevisiae, not Saccharomyces. I'm so used to just being very lazy with my terminology that way. So. Okay, so with that, and I'll put this back up right before we talk about our sensory as well. So if you need to double check at that point, you can. But oh, I should also say, Jenna, I think has probably already put the draft reports into the chat. But if you want a hard copy of the data that I'm about to show, those should always, Jenna's going to put those in the chat when we get to each experiment. So if you did want to look at those a little more up close, you can do that. So the fermentation curves themselves look pretty straightforward. As Matthieu said, the STR seems to be a strong fermenter. It has good kinetics. It doesn't kind of it doesn't slow down at the end and not give you a result. And the Uvarum also seemed to be a good fermenter at this stage. And in this case, the Uvarum does have sort of a temperature range, but this barrel fermentation was well within that temperature range. So we don't have to be worried in this case about getting for Uvarum, it's always getting too warm. So this did not get too warm to stress the yeast in any way.
The barrel were side by side, and just like, I don't know why this variation of temperature, but it's
yeah, I don't know. Well, I mean, that could be part of the yeast activity as well, right? So, but I don't know why they're going up and down and up and down. It might be,
it's
really not that.
We will check the temperature that day if it wasn't the same person,
Exactly. They're reading it a little bit different. There's nothing in here that's concerning, I think is what I would say. So now when this did finish, Matthieu looked at his chemistry in-house, and he was like, my pHs are very high and I would like to add acid now. Can I add acid now? And what I said is, of course you can add acid now, but I would really like to look at the malic acid at that stage before you do any addition, just to make sure that we're not shifting anything about the pH or the malic acid right at the end of fermentation. So we were able to pull samples from each of the barrels for each of the yeast groups, and look at the pH and the malic acid at that point before we got a full chemistry panel after he did his acid addition. Unfortunately, it does not look like the Uvarum yeast, the QRHO, was able to make any difference whatsoever in the pH. It looks just about the same. And if anything, it looks like we have lower malic acid in the QRHO. Now, when Matthieu and I saw this, we were like, well, maybe it is a barrel fermentation of Viognier. Maybe it sort of kicked off its lactic. Maybe there's some lactic fermentation going on there as well. Once we, and we'll see the answer to that question in just a minute. Once this was done, then Matthieu went ahead and did his acid addition. So he added 2 grams per liter of tartaric and 0.5 grams per liter of malic. It got sulfured at that point, and then we could send it for full analysis and have confidence it would make it all the way to the lab in good shape. So when we looked at the general chemistry post-fermentation for all of this, after the acid addition, just like before the acid addition, we did not see any meaningful differences in pH based on the yeast itself. We did see maybe just a tiny bit higher titratable acidity, but if you've ever run TAs, you know that's not a huge difference at all. It's probably not meaningful. It may just be lab artifact. The malic acid, again, does not seem to be any higher. If anything, it might be a little bit lower, in which case that means it might be actually consuming a little bit of malic acid in this particular fermentation. And we did get lactic acid at this point, and there was no lactic acid at all. So that change in malic acid is not due to malolactic. That's really due to what's happening with the yeast themselves. So unfortunately, we did not see that effect of it. However, we did see a couple of other effects. The most notable of which is this difference in the acetic acid. So the STR fermentations, both of them finished fermentation with an acetic acid in the 0.55-ish range, whereas the QRHO finished fermentation with no perceptible acetic acid in the wine at all, which was very notable, I think, in terms of what sometimes we're dealing with in Virginia. We did see just a little bit less alcohol conversion with the Uvarum based yeast. And again, that might just be, we know that yeast have different alcohol conversions, but that might give us a clue that we maybe at least have a little bit of glycerol production. And then the last thing we did see here is a little bit higher total sulfur in the Uvarum versus the STR. I'm not going to talk about that too much right now, but we're going to kind of see that again. So I just wanted to make sure to point that out along the way. So that's what our chemistry looks like. But again, kind of one of the more interesting things about this was kind of thinking about what does that floral character do as well? And do we see any differences in the mouthfeel? So that's where we'd like to go back and talk a little bit about your sensory. So at this point, I think we're going to go ahead and Jenna's going to launch a poll just to ask you 2 basic questions to ground our sensory discussion. Were you able to tell the difference between the wines? So based on your triangle test when you were tasting it blind, could you tell the difference? And then which one did you prefer of the 2? And while you guys are doing that, I will stop sharing my screen so that I can see you when we talk. There we go. There you are. So were you able to tell the difference between the wines? And if so, which wine did you prefer? As we go into the time of discussion, also, please, if you are in a place where you can unmute yourself and talk, we would love to hear your voice and hear your thoughts. If you're not in a place that you can do that, or if your technological setup doesn't allow it, we will also be keeping an eye on the chat. If you want to just write into the chat, we'll keep an eye on that as well. So everybody has to vote. So we don't have, we're not quite there yet, but we're close. Just another minute or 2. Okay, last chance to vote. Okay, Jenna, do you want to go ahead and close that out? See if that works. There we go. And then do you hit share results, Jenna, so that they can see? Can you guys see the results? So it looks like we have about 63% of you all were able to tell the difference between the wines. The STR has a slight edge in terms of preference, but we certainly have have representatives from both. So somebody that was able to tell the difference between the wines, what were the main differences that you saw between the wines? And again, you can go ahead and unmute yourself, or you can write into the chat, either one.
If nobody wants to talk, I'm going to start talking, but
So get
I'll share.
Okay. Thanks, Chad.
I got a noticeable VA aromatic on the STR that wasn't there on the other two. And then also the floral expression on the Uvarum was really evident there for group 1. So 561 for me was definitely the outlier. It didn't really also have much varietal character. Esters were being thrown kind of more for me in the Uvarum. You got more of that thiol expression. Almost got me towards almost like a Sauv Blanc Muscat clone, which was really interesting for those two to pop up instead of Viognier. It threw me for a loop. If I was doing blind, I didn't realize it was BO until the end.
Okay, great. Thank you, Shai. Other thoughts on the differences between the wines? Did you guys pick up the same thing? Did you have different impressions?
So this is Randy Phillips. Same impression here. The Uvarum was distinct. We've been doing Viognier for 20 years, and it was very noticeable. The Uvarum, the freshness, the fruit expression was so much better for me.
Okay. Great. I noticed that freshness as well. I felt like it had a liveliness to it. Great. Thank you, Randy. Other thoughts on the differences between the wines? What about preferences? So somebody that preferred the STR, tell me, why did you prefer the STR? There were lots of you, I saw. There's 11 of you that preferred the STR.
Don't hide. I prefer the Uvarum, so I'll talk when you ask about the Uvarum.
Somebody who preferred STR, tell us what you preferred about it. There's no right or wrong answers here, so it's helping us to think about what the different choices are along the way.
Nobody? Gosh.
Come on. There's like,
I'm sure somebody wants to talk.
I will say, I mean, having tasted both of them myself, I will say that I felt like with the STR, I did get a little bit more of kind of what we would classically think of with Viognier. So a little bit more of that sort of peaches and apricots and nectarines. I felt like that it was sort of more sitting in that category. So it was a little bit more, as I think Shai said that too, recognizably Viognier along the way. So I'll be interested to see that Viognier varietal character number. Okay, so people who preferred the Uvarum, why did you prefer the Uvarum?
Nobody likes anything. In fact, like, the wine was so terrible that nobody wants to talk about it.
Matthieu, I do have a question. So it was a tough year in Central Virginia to ripen Viognier.
Yes.
And what was
I don't know why you're saying that. Is because like I wasn't able to push it more than 21 breaks.
Well, just I know we were certainly struggling on how far to try to ripen Viognier, just because the weather wasn't there this year for it. We were getting a lot of rain, so our wines are definitely less expressive than in a riper vintage. So I was kind of curious if you were looking for a way to coax more aromatics through the yeast out, or if this trial was kind of already set before the vintage played out.
Oh, it was already said before we started the vintage. I had no preconceived idea about what I'm going to get. I was definitely more interested into the acid component of the wine than the flavor profile. The flavor profile for me was an afterthought. It was like, yeah, let's see if I can correct my acid. Even if it's too floral, I don't care. And as like, a lot of sales reps, they'll sell you anything, telling you it does exactly what you want it to do, and it's not the truth. So, you know, it didn't change at all my malic. It, in fact, depleted my malic instead of increasing it, and didn't change my pH. I've got almost higher pH on the uvarum. So, but still, to this moment, I personally prefer the uvarum, even if, like, for me, 0.5 VA is not high for white wine. I mean, it's not low, but it's not. I'm not too worried with the barrel fermentation at 0.5. I'm like, yeah, it's okay. I still taste it, and that's kind of like bother me a little bit on the STR, especially when I compare them side by side. I never do my barrel fermentation in my Viognier. It's only I would say less than 30%. It's like probably 25% of my blend. It's a barrel fermentation. Everything else is tank fermentation. So even if I've got a bit of VA or higher VA perception, I will say on my barrel fermentation, I'm not too worried about it. It's going to be blended with my still wine, my stainless steel wine. And even here, if I've got more floral component, and maybe it's not like air quote typical Viognier characteristic, I still think that the flavor profile that it brings is super interesting. I like slightly more floral Viogniers than the overripe, apricot peach style of the Viognier. So I think that fit well into my flavor profile that I'm targeting. And I really like the freshness. And very often I feel like the Viognier can be too soapy, too heavy. I like the brightness and the freshness that it brings. And in some weird way, I also don't dislike having a very, very low VA on this wine. So, you know, not that I'm obsessed with numbers, but at least I don't perceive any VA. So.
And with your barrel fermented Viognier, how often are you stirring? Is it just before pulling samples for a WRE trial, or more frequently than that?
No, just I don't do much on my wines anyway. So, you know, it's really just to keep. I don't steer the Viognier. I steer the Chardonnay. I've got a true regimen steering on my Chardonnay, but on the Viognier, I don't feel I need much more manoprotein from the lees. I feel like you know, again, I'm not looking too much of the fatty structure of the Viognier. So I do feel like usually my barrel fermentation Viognier brings me more of the peach and apricot component compared to my tank fermentation that brings me more ester. But I don't know. I like the uvarum better, and I'm probably going to keep using some at least and see how I can play with it into my blend.
Yeah, cool.
Okay, thank you guys. So any other comments about this set of comparisons before we move on to the next one? Any other comments about Matthieu's Viognier?
I want to try it on Petit Mansang next year, because I want to see how it's slightly diminished the alcohol on the Petit Mansang. Okay. And I want to see how this like very estery component will play with the pineapple of the Petit Mansang.
Oh, that's a good idea. I think we were afraid to do it on Petit Mansang this year because the data that we had from the in-house trials had this like big increase in malic acid. So we were kind of expecting or trying to work around that. At the end of our time today, I'll share with you the results that Tyler sent to me from some other trials as well this year. So it seems like we can maybe control that malic acid a little bit. If we're not seeing it in this one at all, in order to be able to make it a little bit more approachable to do on Petit Mansang. So I'll definitely sign you up.
And also on the Petit Mansang, especially on the barrel fermented Petit Mansang, I've got a tendency to have much higher VA. So that I want to see also if it's got any impact on the VA on the Petit Mansang. So.
Yeah, I think that's a very good point, is that those barrel fermented Petit Mansangs can finish at 0.6 pretty easily. And sometimes the Viognier can carry it, but you know, how would it
low for me? That's okay.
So I said easily, sometimes even more. So, okay, so let's go ahead and move on to our second flight. The second flight was an experiment done by Rebecca at Cunningham Creek. And so I'm going to go ahead once again and share my screen so that we have some visuals to go along. Rebecca, why don't you tell us just a little bit about why you were interested in what your experiment was and why you were interested in doing that experiment?
So for pretty much the same reasons, we always struggle with really high pH in our Viognier. And so anything that can help us add less a commercial acid would be great. And also just, for cost reasons. But we also, when we add acid to fermentations, we usually do a ratio of tartaric to malic. So we obviously didn't add the malic in this case, but I just wanted to see if what we're doing is actually worthwhile or if we should just be adding tartaric anyway. So if that could, shine some light on our acid additions as well. So we also did 4 barrels of neutral French oak. Our juice actually started fermenting in the tank before I was able to barrel it down. But our first 4 barrels were still, since there was a gradient in the tank. And so when we inoculated with the Q row, we just kind of assumed a co-fermentation with a natural yeast.
Right. Yeah, and I think that this is actually an interesting, it sort of was unintended, but it's kind of an interesting wrinkle into the discussion in terms of kind of how does Q row work with other yeasts around when other yeasts are around, because most of the time other yeasts are around, it's just a matter of how much. So as Rebecca said, this was initially planned to be kind of exactly the same as Matthieu's, with cold settling and then racking, homogenize, and then go into the 4 barrels. But that cold settling had to happen for a little bit longer than expected, and it did start to do a little bit of fermentation. So in this case, when it went down into barrels, 2 of the barrels got inoculated with Q row, and 2 of them were just left to continue their ambient fermentation. There is a little bit, we kind of felt like this was an interesting question for 2 reasons. Enartis did initially give us some instructions on co-inoculation. So if you were trying to kind of moderate some of that rose character or make sure that you could still go through mallow later, they were like, it's better if you co-inoculate. So we sort of had that. There's also one paper in the literature kind of looking intentionally at how does uvarum compete when Saccharomyces is around. So this particular diagram basically just what it's showing is the proportion of Saccharomyces cerevisiae, which is the darker gray, to Saccharomyces uvarum, which is sort of the unshaded portion of the column, depending on inoculation ratio. So the third one down, which is marked C, that's at a 50-50 ratio. You can see that at warmer temperatures, cerevisiae takes over. At cooler temperatures, you get uvarum kind of hangs around at about 25%. But then D in the upper right-hand corner, that one shows you what happens when you have sort of a 10-to-1 ratio of uvarum. Uvarum holds its own against Saccharomyces cerevisiae. And so we felt like in this particular experiment, we had enough to kind of say, okay, we're still going to see if we get some uvarum character, even though we've got some cerevisiae happening along the way. So with that, we did, again, we took once the juice went down, it was into 4 barrels, but the setup there then was ambient fermentation and then the Q row into other barrels. We did look at the juice chemistry right then. And again, you can see we're starting at that really high pH again, which is just confirms to us that, Viognier is a good option for this particular experiment. We did take more chemistry, but it basically told us the same thing. So
yeah, and
so that you know what you have. Sure.
We did add some tartaric at the initial barrel down, just because with the natural yeast, I was a little concerned about, making sure it completed and giving it the best start so they wouldn't get super reductive.
Yeah, so and that would be kind of the normal kind of best practice would be to do an acid when you have that high pH to do an acid addition. But in that case, the acid addition was the same to all 4 barrels, and so we feel like particularly our malic acid, comparisons are clear later on. so that you can see what you've got in front of you. Again, remembering that we're kind of assuming that the ambient fermentation was Cerevisiae. In most cases, in warmer regions like this, that would be a Cerevisiae, particularly when there's a lot of other commercial Cerevisiae around. That's a fair assumption to make. So those are the wines that you have in front of you. So Rebecca, tell us a little bit about how fermentation went.
So it went pretty well for the inoculated barrels with the Q row. The ambient ones, I don't know if it's just because, as I said, there is a gradient in the tank, and since we pulled from the bottom that had little to no fermentation activity to start with, if it just, you know, didn't have enough yeast or whatever, but it had to be restarted beginning of October just to get it through, because I was worried about it going through malolactic fermentation. But otherwise, the Q row did exactly what it was supposed to. It did finish with a little bit of residual sugar, which will be blended out, so it's not that big of a deal. I ended up stopping it just for concerns about once again, the natural yeast and completion purposes.
So yeah, so when you kind of look at these fermentation curves, they're pretty standard. They're in a lot of cases what we would almost expect to see for an inoculated fermentation versus an uninoculated fermentation. It looks like you have a little bump point there, so maybe something happened at that point, but a lot of uninoculated fermentations do take a lot longer to get through. And, you know, I think it is an interesting comparison in that, you know, we can think about Q row as something that, you know, can it overcome the natural yeast or not? So in this case, it does look like it's kind of overcoming what's happening there. When Rebecca called and said, you know, we are going to sort of restart these fermentations, we did once again sort of pull some chemistry at that point, just in case there's anything about the reinoculation that went strangely. So at that point in early October, when we looked at the pH and the malic acid of those two wines, once again, we did see that the Q row really did not help the pH at all. If anything, it was a little bit higher, and the malic acid was a little bit lower. Now we had that same response, and maybe even more so here, as we did with Matthieu's, that, you know, that malic acid might be partially due to some malolactic conversion. And when we know we have some ambient ferment going on, it's likely that we've got some malolactic going on as well. But at that point, we were at least able to kind of capture that number. And then afterwards, we were able to look at the finished wine chemistry once things were all finished out and we could get some sulfur in there. So the finished wine chemistry, once again, the pH looks exactly the same between them. So we don't see any benefit to the pH of using the uvarum yeast. Once again, the TA is just slightly a little bit higher. And again, I don't know if that's meaningful or not. This is a little bit more of a difference than what we saw in Matthieu's. And there is one, uvarum is known to make malic acid. It's also known to make a little bit of succinic acid. So it might be that we're making a little succinic acid, not really affecting our pH, but having a little higher TA. But the TA can be affected by so many different things. It's really hard to kind of hypothesize what that's about. Once again, we see a little bit less ethanol conversion in the uvarum versus in the ambient fermentation. In this case, that ambient that was restarted did finish all the way. The uvarum, one of the barrels finished and one of them did not. You are tasting the one that did go all the way through fermentation. So you're tasting barrel number 3, just so that we can have a good comparison there. I will say at this point, some of the stuff in the literature with uvarum does indicate it might be a glucophilic yeast, meaning that if you may end up leaving some residual fructose along the way. We do have some bottles of this left over, so I'll probably send, particularly the 4th barrel, we'll kind of send it to see if that's indeed fructose or if that's glucose that's left over. And then once again, we have much higher levels of total sulfur in the uvarum versus the ambient fermentation. So again, I don't know what that's necessarily about. We're just recognizing kind of, in this case, we've got 2 of them that both have that happening. That's not something that's showing up right now in the literature, but it's just something to keep an eye on along the way. So that's what's happening with the general chemistry. Then thinking more specifically about what's going on with that malic acid. In this particular case, we did have some malolactic happening. So this graph shows you the blue portion of the column tells you the malic acid that was in each one of the barrels. And then the orange part of the column tells you, I'm calling it malic acid equivalence. So this is lactic acid that has been back calculated to if it were malic acid. So we wanted to see, did we sort of start with more malic acid to begin with? And of course, during the course of malolactic fermentation, it does throw off a CO2. So there's some calculation in order to sort of get those to be equivalent numbers. But with that calculation, what we can see is there's really no meaningful difference in the malic acids at all. So whatever's going on at Enardis's lab does not seem to be happening in our barrel fermentations in these two experiments. And then lastly, we want to look at the acetic acid. So if we look at the acetic acid, we had 4 different barrels. Our 2 ambient barrels did have higher acetic acid than the inoculated one that sort of went through without problems. The one that did have a little bit of residual sugar, its acetic acid was a little bit higher again. So this is kind of inconclusive in terms of, do we have a little bit, would we see the same thing if we were comparing 2 different inoculated fermentations? Is that a little bit higher acetic acid due to just the fact that it's ambient, which we know happens? Or is the higher acetic acid in barrel number 4 just because it probably had some issues at the end and maybe struggled a little bit and accumulated some. So this is kind of inconclusive. It might sort of be along the same trend, but inconclusive relative to what we saw, with Matthieu's experiment. So once again, though, there is sort of a big sensory component to this. So at this point, I'll remind you of what you have in front of your glasses, and I'll ask Jenna to go ahead and launch the second poll so we can start our discussion of the sensory here. Oh, looks like we have everybody. So yeah, Jen, if you want to go ahead and end the poll there. It does look like it was very easy to tell the difference. Many of you were able to tell the difference here. And so let's talk about that first. I think, tell me what were the differences you were able to tell between the wines?
For me, that the ambient was more aromatic and had was a bit brighter, so I preferred it for those reasons.
Okay. Other folks, what was the differences between the wines? There seems to be a stark difference. So sometimes that makes me think there's something.
I think that I was picking up a little bit of diacetyl production on the ambient. It provided a nice fatter characteristic to the Viognier as opposed to the clean acid-driven notes from the Uvarum. Uvarum, I mean, it's going through for the Viognier panel that we're doing right now. It's holding true. So like you get the similar aromatics from both of these Viogniers for that floral characteristic, which is really shining through in both of those. I was in group 1.
Okay. Great. Other differences between the wines or preference reasons we have preference. So we've got, yeah, so far we've had 2 folks that prefer the ambient, but we do have a number of folks that also prefer the Uvarum. So somebody want to speak up for Uvarum here?
I actually like the ambient a little bit better. I'm kind of on the fence about preference for both of them, but I thought, like Mike said, the ambient was brighter, and for the Uvarum, I got a little bit of a spicy characteristic, and at one point I did get VA, but that sort of went away. So both samples were fine. I think they were varietally correct. So that's my 2 cents.
Great.
Yeah, I mean, I prefer the Uvarum one. I do really like the floral quality to it, and I felt like the ambient, that roundness was a little too, I think people are gonna think it's buttery, which may, in the struggling fermentation, I could have had some malolactic conversion. But yeah, I mean, I like the potential reduction in acetic acid, and even though it didn't really do anything for our pH, those barrels were easier to adjust pH on, if that makes sense, when we did our final ones, than the ambient ones.
Okay, so you feel like you got more for the same ad?
Mhm.
For me, I mean, I obviously I prefer the Uvarum. I think it's cleaner than the ambient. For me, the ambient is a bit too funky. But there's one thing that on this one, I mean, I feel like the pH of both wines are very high, but like I didn't care much for the mouthfeel of the Uvarum on this trial, because I feel like it was very narrow and very acid driven, but not in. I feel it was missing some flesh. So I do think that the blend of both of them will work good together. I was missing flesh on the body and flesh on the Uvarum, but I do like it's more straight and more clean. So I like that better.
And I feel like that's interesting, Matthieu, because remember when you and I tasted your two sets of barrels, we felt like one of them was sort of the mouthfeel was not consistently different between them. So one of them we felt like there was kind of the Uvarum was a little bit more pointy, and one of them we felt like for you the STR was a little bit more pointy. So that may be one of those sort of mysterious, like what's causing that shift between production of ethanol and production of glycerol. And it might just be one of those kind of biochemistry things that is not entirely clear yet here for the Uvarum. And I want to go ahead and bring up too, Tom has written into the chat chemistry question about how the RS on the last QR batch was 12.1, and yet the alcohol was so close to the others. And Tom, I don't really know. I haven't really looked so closely at that yet, but it's a good question to say, why do we have such different, we would expect to have even lower alcohol on that. So I'll look back at it and try to about calculate it and see if sometimes it's just sort of the range of error in alcohol testing, but I haven't back calculated it. So thank you for pointing that out, and I'll try to back calculate it and see if it makes sense that way. So.
Joy, for me, the ambient was not classic Viognier. There was something in there that was really masking what I would expect from a Viognier. Uvarum, they're classic Viognier. It's just my personal preference.
Yeah. And I think it is an interesting, like, again, that both of them in this case had gone through a little bit of malolactic, but it seems like people were picking up more of that lactic character on the control than they were on the Uvarum. And that might just be because of the sort of those other components of having different yeast around, or even that kind of extra ester production that we find from the Uvarum really kind of pushes it to what we think Viognier should be. So the question would be, how would that show on different varieties that maybe don't have that same characteristic as as one of the varietal characters? But
yeah, we did a barrel of indigenous yeast this year, so I'm anxious after this is over to go out and taste it and see what it's like.
Yeah, it's always an adventure when you stop trying to control the microbes that are happening. So okay, any other comments on this experiment before we go on to our third experiment for the day? Michael?
I was curious. I noticed that the Brixit harvest was at 19.7, but I knew there had been some fermentation activity. So I would, and also the wine feels quite ripe to me. So I was curious if there was any chaptalization done, or if the 19.7 had already kind of fermented a bit. So
Yeah, so when we took the juice, I took a juice sample before we did any additions at the barreling. It was actually, the ambient ones were, it was a range between 20.4 and 19.9. So the Brixit press, I don't know, but it could just be a reading error. We just have one of those like little eyesight things, which, you know, has a lot of human error involved. But I did chaptalize with 30 grams per liter of sugar, just to get it up there.
Cool. And the other thing is we saw the same trend with the lower alcohol in the uvarum than the ambient or cerevisiae. Just I don't know if anyone else has had this experience, but I tend to see better alcohol conversion in an ambient fermentation. So I was curious if that difference was even more pronounced in this trial as compared to Matthieu's trial.
Well, let me check. I have a good question. Let's see. And I think Jenna has, if you want these raw numbers for yourself as well, Jenna, I think is putting those PDFs into the chat as well. So hopefully those
go
are
ahead and
showing.
send those.
Yeah, go ahead and put the PDFs into the chat so that they can see. It does look like that difference is, in Matthieu's, it was only a small difference. It was 0.2. And then in Rebecca's, it was like 0.4-ish, depending. So yeah, it was a little bit more. And Michael, I would say that, you know, sometimes that alcohol conversion and the sort of slower, cooler ferments that we get for our ambient ferments does, I would concur that I've often seen that greater alcohol conversion. And I've always thought it was heat, but it certainly can just be the yeast themselves as well. So yeah, that's a good point to bring up. And just the difference in the bricks at harvest versus the bricks that the lab came back. Sometimes that's just as Rebecca said, a difference in the instrument. So reading with the refractometer is usually going to give you a little bit lower number than reading with a hydrometer, and it's just part of the background of how those two things are working. So so that's all that difference probably is. So okay, any other comments about this experiment before we move on to our third one? Okay, so our third experiment was done by Rick Tagg at Delplane. So Rick, you want to tell us a little bit about what your experiment was and why you wanted to do it?
Okay, well, our experiment was on Petit Verdot, and we have a block of Petit Verdot, just about an acre, at a satellite vineyard on Leeds Manor Road in Markham. And basically Joy told me that I had to do it, so I did it. But it was a good experiment to do because I'm always concerned with high pHs and VA. Especially when this one, it tends to, this particular vineyard, this particular block of Petit Verdot tends to get ignored to the last minute traditionally. So we've kind of focused on it this year. It actually was the last lot of grapes that were picked. I think they were picked October 23rd. Joy, you have all the numbers for everything, right? So
Yeah,
that goes on with that. But I think that my idea was to see how it affected pH. My idea from the evolution of seeing everyone else's experiments is the VA potential for that, because that's my latest obsession. So that's about it. That's really about it. I will say that when we did do this, it was like I say, it was the last grape picked. It was really late into October, and the co-inoculation and the math to figure out how much of the D254 was the other yeast to use in comparison to the experiment, the Q8 Archo or whatever it is, really hurt my brain at that moment. So by
Yeah,
doing
it
this
is.
again, if for those of you that do these experiments, I think that it's probably a better idea to do it with an early varietal. And Joy and I talked about that because there's a
Yeah.
tendency, especially, and now Joy, we ought to also talk about juice samples at some point too, because it does come up that for our labs that we use, our sampling for our juice samples, if we can't get them to the lab right away, or we don't do it in-house, is typically those juices are frozen, which you're going to get a different reading on different things from the thawing out process, or whether or not that sample thaws out in transit. So one of the other things that I learned that was really important to me, and I'm actually going to work into my harvest parameters this year, is boiling the juice before I send it off, so that it won't ferment or it won't have the potential.
Yeah. So, and that's a good point, Rick. And I think that's
If I can't get it to the lab right away.
Yeah. So it is a good point that when we're taking our juice samples, and we'll see Rick's juice samples in just a minute, but many of you are kind of taking these juice samples, but you're not able to get them to the lab that day. So they end up either in the refrigerator or getting frozen. And that does sometimes shift your pHs and your TAs in particular.
And your sugars. So yeah.
Yeah, exactly. So if it starts to ferment, it can start to do that. So what we employed with
Yans too.
Yeah.
So, yeah.
Yeah, if it starts to ferment. So what we employed with Rick's experiment is we did do kind of a small boiling, but the one thing you do need to be careful with if you're boiling it is you need to make sure that you don't that you replace any water that's been evaporated due to boiling, because that changes the concentration. So the way that you do that is you weigh the container, then you do the boiling, you weigh the container again, and any weight that it's lost, you replace it with distilled water. So if anybody wants to discuss strategies to get good lab samples, please let me know. Everyone sort of has their own situation. So I'm happy to talk with anybody about how to try to do that. So getting back to Rick's experiment here, though, of course, Petit Verdot is one of our really high pH red wines, and it seemed like it was a good candidate if we were going to try Uvarum on a red wine, which at the time they were telling us it could work on reds or whites. It seemed like Petit Verdot was a good candidate for that. In Rick's particular experiment, he had about 1.3 tons that came in. He destemmed that equally into two different bins. And as he said, one of those was inoculated with D254. The other one was inoculated with kind of a 1/3, 2/3 split. So 1/3 D254 and 2/3 of Q-Row. And there were a couple of reasons that we did that mixed inoculation. And as Rick said, it did turn out to be the sort of mixed inoculation is logistically more difficult because the yeast have different thermal needs during their rehydration. But the reason we did the reason we did the mixed inoculation was that at the time there was some question about whether this yeast was really compatible with malolactic fermentation. One of the studies that had been done through Fresno State that an artist had sponsored the year before did have some trouble getting through malolactic. So Joshua, who was in charge of the trial at the time, recommended that we sort of do this split with the hope that whatever was getting in the way there wouldn't get too much in the way. The other idea was that some data had come back that it was glycophilic and was leaving some residual sugar. So the hope was then that your Saccharomyces cerevisiae yeast could come and kind of consume any sort of help out to make sure that you're not leaving fructose behind. And then again, that sort of high rose character, we didn't know if we kind of wanted that to run amok in the Petit Verdot. There is a floral component to Petit Verdot, but not the same way as that we would see with Viognier. So again, the recommendation from an artist was to do this mix. And their feeling was that even though it's a cryo-tolerant yeast and it tends to sort of compete best with cerevisiae at cooler temperatures, all of our ferments start cool and then they get warmer. So the idea was that you would sort of get some of this at the time, we were looking for malic acid production. We would get some of that on the early end of the fermentation. And then essentially our cerevisiae would kind of take over later in the fermentation. So the idea is this a good co-inoculation yeast, all one inoculation, but you kind of get best of both worlds along the way. But as Rick said, it does sort of require a little bit it's a little bit tricky to get all of that rehydrated and calculated and everything to do your inoculation. It is only
Meant for
one.
timing, it's a stage of
Yeah,
timing.
exactly. Yeah. It's a little bit, I think it's a little bit less logistically difficult than like if you're doing a non-Saccharomyces yeast where you have to remember to come back 2 or 3 days later and get your inoculation at the right time. But there is a little bit of calculation that's needed along the way. So that you know what you have in front of you then. Again, we had our D254 was our Cerevisiae yeast. And at the same time, the Q row is still our Uvarum yeast. Same yeast, just very different application in this case. Okay, so when we look at the chemistry for the juice coming in, you know, this is that trick of Petit Verdot, right? You look at this pH and you're like, why would I ever have to add acid to that? But those of us that know Petit Verdot understand that that is a very deceptive number right there. So it is something that we'll see in just a minute. There was certainly reason to add acid if we wanted to. The fermentation seemed to progress very well for both of them. In this case, though, the Q row was actually a little bit slower on the uptake than the D254. This is a little bit counterintuitive. I think that the hypothesis would have been that the Q row would actually get started a little bit faster since it does better at cooler temperatures. That just didn't seem to be the case in this particular circumstance. And again, some of that, from talking to Tyler from Vineyards, some of that might have to do with the sort of rehydration itself. So Tyler did come back after this year and say, you know, they're finding that it is a pretty delicate yeast during rehydration. And if you don't kind of get every little step exactly right, you kind of lose more of your yeast along the way. You don't end up with as strong an inoculant as you think you have. The general chemistry, again, we finished with a very high pH, so there was no worry that we didn't need to add any acid along the way. But again, in this case, we don't really see any benefit from using the Uvarum. We did take our malic acid number. I'm sorry, I should say this is pre-malolactic. So this was when Rick just took the free run of these and put them in his barrels for malolactic, and we went ahead and pulled a sample there to try to get a measure on what the malic acid was going to be at that point. As you can see, the malolactic had already started to kick off. So once again, we sort of did that calculation to malic acid equivalence here. And it does look like with malic acid equivalence, we do have a slightly better return on our malic acid in the Q row versus the D254. But of course, we'll look in just a minute at when it's been all converted and see if that's consistent or not. In Rick's case, the acetic acid at this stage was still very manageable, and there weren't any differences between the two. And again, and we don't really see much difference at all in the ethanol conversion. When we look at the numbers post-malolactic, mostly it looks the same as what we saw pre-malolactic. It just sort of holds that our pH is pretty much the same. We do have a little bit more titratable acidity in the co-inoculated wine. And once again, we do see we have more lactic acid than we do in the D254, which indicates to us that we did build a little bit of malic acid. When I did the math on that, that was like, you know, about 20%. It's not huge, but it is something. And again, in this case, the acetic acid is slightly lower. None of these acetic acid, this acetic acid for a red wine is very well in line with what we would expect, you know, or even hope for. So there's no concern with either one of these. And the question is if Rick had a higher acetic acid, would the Q row would have shown a bigger difference or not? And again, here we don't see a big difference in total sulfur. So some of those trends that we saw in the Viognier, we're not really seeing as much here in the Petit Verdot. But again, one of the big questions is, what does that mean in terms of sensory? So just to refresh your memory on what you have in front of you. And Jenna, if you could go ahead and launch that third poll for us. So same questions as we had last time. Were you able to tell the difference between these? And if so, or even which one did you prefer of the ones that you had? And with that, I will just a couple more people to answer there. Last call for voting. Okay, Jenny, you can go ahead and stop the poll there. Oops, I missed it. There we go. So it does look like, it looks like this one was much harder to tell the difference. We had very few people that were able to tell the difference between the wines. But once you knew the difference, we still had preferences because we made you have preferences. So somebody who was able to tell the difference between the wines, you know, what if anything was the difference, or did you just happen to guess well?
So a little more difficult. I'm really pleased though. I got all three of these right. I rarely do that. The Uvarum for me was the more classic Petit Verdot. The other was a little muted, but it was very, I mean, very subtle. It was very subtle.
Okay. Was there a particular characteristic that you felt was muted? Was it the nose?
So for me, Joy, one of the flavor profiles of Petit Verdot is an olive, and I tend to get more of that with the Uvarum.
Okay. Okay. I get that. I feel like D254 is often very like kind of berries and fruit and that kind of thing, and maybe not as much of those others.
Yeah.
Okay, great. Thank you, Randy. Other differences that people notice between the wines?
It was very hard for me to tell at first, especially driven by the taste of the wines. I made my distinction based on the finish. I felt like the Uvarum had a more smooth finish versus like a lot of tannins, which you get with Petit Verdot. So that's where I made my distinction, even though flavor was very, very similar.
Okay. Yeah, and that's a good point. And that might go back even to if there was a little bit, for example, of glycerol production, that might be sort of moderating some of the tannin finish a little bit. Or it may just be, you know, the effect of some of those aromatics kind of filling it out for us. Okay, thank you. Any other differences between the wines? What about preferences? So we've heard a little bit of, so Randy kind of told us a little bit about what his preference was. Other preferences between these two?
If we just, I mean, for me, I failed on this one, so I didn't detect any difference. Either I'm a bad taster and that's possible, or there's really not that much difference between both of them. But to my point, I did click because I didn't have any other choice to click one of the option, which one I preferred. So I did click D254 because if I don't taste any difference, I don't see why I will be using Uvarum to get a wine that I feel is very, very, very similar. So I don't see, and even now when I look at everything, you don't see a lot of difference in the numbers. You don't see a lot of difference in the flavor profile, maybe a little one, but even going back to it, I don't taste it. So for me, it's like, you know, why adding another product if you don't taste the difference?
And I think that's an excellent point, Matthieu. And just to sort of clarify there too, you know, it's an important thing to know if there's no difference. So you didn't, you know, if you can't tell the difference between the wines in a triangle test, that's really important information for us to know, to say, you know, we're trying to determine, is this yeast even expressing in this sort of 2/3, 1/3 mixture? And it may very well, from the chemistry, it doesn't really look like it's doing much. And if the sensory is also not picking that up, it may just be that the Cerevisiae is kind of out competing it here. It is a relatively warm fermentation. There's lots of things about it that we would think maybe the Saccharomyces cerevisiae is, you know, kind of taking over in that regard. So it is an important thing that we kind of recognize if there's not a big difference. And that's where your sensory sheets become really important as well. We'll be able to kind of determine if it was, you know, if there's meaningful differences there or not, at least in the group that taste it. So So before we wrap up today, any other comments about the sensory portion for the Petit Verdot? I do have a couple of kind of summary comments at the end here, but I want to make sure we get everybody's impressions.
My internet keeps going in and out, so
Okay.
anyway, I think that, like with all red wine yeast trials, at the beginning of the trial, there was more of a distinct difference in the wines. And as time has gone by, the wines have gotten to be more similar in how they've tasted, for the Petit Verdot in particular. And what I got out of it was actually how much I appreciate D254. So I think that it made me actually focus on what the characteristics were that I was actually looking for. And the anecdotally appreciating what the D254 brings out in the fermentation really helped my planning for this coming harvest a lot. So that's my 2 cents.
Yeah. And Rick, I think you make a really good point, is that with reds, some of these aromatics are a little bit more sedate. So Shai, did you have something you just put your hand up?
Yeah, I noticed that, did I see that right? That after post-mallow, that the Uvarum actually went down in pH from 4.06 to 4.03 versus the D254 that actually increased from 3.9s to above 4. Like that's impressive too, if we're looking at a reduction in pH or a reduction in our stability for our reds.
Yeah, I mean, I think in that case, that difference between 4.06 and 4.03, those were 2 different labs that took that number, and that's not a big difference in pH. So I would feel like that's relatively the same. The
Post ML though, like we're looking for an increase in the pH.
yeah.
So if it was just a straight comparison, I see that wouldn't really be that much of a difference. But if it's after ML,
yeah, I mean, I would say that there were different stages of ML when we took those numbers. So I would think that those finished pH numbers would probably be more a better gauge of what it was actually doing, just because, you know, one of them had gone further into ML than the other had, and so probably one had affected the pH more than the other one had at that point. So I did want to share just a little bit of an update from Enardis. So I spoke with Tyler from Enardis about this, and as I said, he will be talking at Eastern Wineries. And so, well, if you are going to be at Eastern Wineries, there'll be more information about it. But he did give me a few bits of updates from some of these trials sort of that have been happening. Of course, we did this trial, but also they had some going on in California as well. Let me get this here. So some of these things are things that kind of line up with us, and some of these are sort of preview of things to come. So they are also seeing this decrease in acetic acid, and we were not necessarily measuring the acetic acid on the juice coming in, because we were looking mostly for malic acid. But what they're seeing is even juice that's coming in with higher acetic acid, they're seeing it lower during the course of fermentation. So that does seem to be something that is occurring. And he did say with all of these things, the more you start with, the more effect that you see. So if you're starting with high VA juice, you're going to see more of an effect. The malic acid production does seem to depend a lot on the amino acids that are present. And there is at least one paper in the literature right now looking at, if we feed it different amino acids, what does that mean for what it's going to pump out the other side? And there are particular amino acids that tend to go into this metabolic pathway that makes malic acid, where other ones don't necessarily do that. Unfortunately, there's not really clear guidelines right now. If I'm going to try to make malic acid, do I need to feed it a particular nutrient at a particular time? That data is still forthcoming. He also said that that's sort of linked to any malic acid production is linked to really high pHs starting, but the number he gave was 3.7. And we certainly had, plenty of both of our Viogniers started with higher pH than that, and we did not see it. I think the difficulty with needing to feed for this in Virginia is that a lot of our juices come in with sufficient nutrient already. And so we're sort of not wanting to have to feed more than we need to. And then he also sort of reiterated that it does seem to be, this temperature thing does seem to really matter. So in terms of the fermentations themselves staying cool, but also that rehydration being at a much cooler temperature that we're used to doing. And he also said that the floral aromas really come out between 40 and 65 degrees Fahrenheit, but more than 80 degrees Fahrenheit, not only will they not come out, but if you have them, they might blow off a little bit, which may be part of what we saw in Rick's experiment along the way. And then the last thing is that this is a yeast that does not carry the killer toxin. And so if you do have any cross contamination from yeast strains that do carry the killer toxin, it will kill it. So D254 is a neutral one, so it's immune to the killer toxin, but doesn't carry the killer toxin. But probably there are yeasts in our wineries, other commercial yeasts that we're using that do have that. So if you're thinking about employing uvarum in your winemaking, there are some of these guidelines that hopefully will become more clear as time goes on. I'd be happy I'll of course pass them along when I hear about them. But if you are thinking about using uvarum, it's probably worth it to talk to the rep for Enartis or myself in order just to make sure that you're getting the most out of your uvarum experience. So with that, I will go ahead and make sure that we do thank the Virginia Wine Board for all of the funding that they give us and the support that they give us to do this. And many thanks to Matthieu and Rebecca and Rick for doing these experiments and being willing to share all of them with us. We are trying to kind of finish the reports in a timely manner and getting them on the website. We probably will just send you a link of these final reports once we have the sensory all completed. But at any point, you can, of course, always look at the website for any of the kind of back information that we've got. So thank you all so much for your time and attention today. It's always great to see some new faces that maybe can't always come to our in-person sessions. So it's great to see you all. And again, just keep an eye out in your email for the link for the registration for the March 8th session at the AREC and the March 22nd session at Stone Tower. I'll happily stay on with more questions